Greetings everyone out there. With the continuation of previous class, here also one more class related to the important plant pathology questions asked from agricultural exams from previous. Here, the method adopted for the assessment of downy mildew of grapes is the answer will be assessment using descriptive charts. Why we didn't choose for this in terms of percentage means, so we have to choose this option only when they have given related to viral disease or related to wilt based diseases or any phytoplasma diseases. Okay, so such type of diseases only we can opt for in terms of percentage. Otherwise we can always use to choose the assessment using descriptive chart only, 0 to 10 percentage, 10 to 20 percentage. Okay, so that type of descriptive chart only will be using for the assessment of diseases. Okay, and the next question is match the list 1 and list 2 and select the correct answer using the codes given in the option. As I already said, they might definitely ask the common name as well as the scientific name of the most important mushroom related. Okay. So here also they have paired with common name along with the scientific name. Okay. So here honey mushroom, its scientific name is Armillaria amelia. Sacred mushroom, Psilocybes emilanciata. Pine mushroom, Trichoderma matsuke, and shiitake mushroom is Lentiononas edodes. So the correct answer will be option D, 2, 4, 1, 3. Next, here the important question, one of the important question related to chemodiagnosis, that is detection using the chemicals. Okay, so I'll read the question first. Banana bunchy top virus can be detected by the following chemicals: iodine test, tetrazzolium test, ooze test, Koch posted. Here you can eliminate ooze test because ooze test is related to the bacterial identification. But here they are asking banana bunchy top virus. So here itself you can eliminate the C option. Then D also you can eliminate because Koch posted. Koch posted is related to pathogenicity test only. So here the detection will not be related with Koch posted. Okay. So C and D option you can directly eliminate. And here you have only two options, either iodine test or tetrazzolium test. Okay. Not only the banana bunchy top virus, some other viruses which affects the banana, like banana bunchy top virus, banana black mosaic virus, banana infectious chlorosis, all the three banana viral diseases can be detected using the tetrazzolium test. One more virus also can be detected using the same tetrazzolium test, that is cassava mosaic virus. Okay, so the most important viruses related to banana can be detected using the tetrazzolium test. And what about the iodine test is? It is related to detection of rice tunguro virus. When the question is about rice tunguro virus can be detected by, the correct answer will be the iodine test, since they have asked banana bunchy top virus can be detected by. So here we should choose the tetrazzolium test apart from the banana bunchy top virus, banana black mosaic virus, and banana infectious chlorosis. So all the three banana viruses can be detected using the tetrazzolium test. And one more also tetrazzolium test you might confuse with the you in the seed pick related question also, using the seed viability also can be detected by tetrazzolium test you might have here. So here the same tetrazzolium also you can used for detection of virus in the banana. Okay, next, the next question, identify the incorrect answer with the pertinent in Ascoma produced by the fungus. Here you must observe here the terminology Ascoma because it is related to Scomicota. You might be knowing there are four important phylum, okay, in the plant pathology related to the fungal classification: Umicota, Ascomycota, Zygomycota, and Basiomycota. You might be here, okay. So here Ascoma, which represents the fruiting body produced by the Ascomycota, okay. Here, when you see the options given, bracket, perithecium, apothecium, and pseudothecium. So all these B, C, and D are three are related to the Ascomycota, uh, that is fruiting body produced by the Ascomycota. Okay, especially sexual fruiting body produced by the Ascomycota. But here they are asked for incorrect answer. So the answer will be the option A, bracket, because bracket is more related with the Mycota. Okay. So the sexual fruiting body produced by the Mycota is also called as bracket. Okay. When we discuss little more elaborately, there are 4 different types of fruiting body produced by the Ascomycota. Naked, when it is completely closed sexual fruiting body, it is called Clistothecium. Closed ascocarp with a small opening at the top, it is called Perithecium. Open or saucer shaped ascocarp is nothing but Hypothecium. So the thecium, okay, so they are all more related to the like not more related, they are obviously related to the Ascomycota. Okay, so sexual fruiting body produced by the Ascomycota are only 4 in number. And the next one is Bestiocar, which is sexual fruiting body produced by the Mycota, which contain Basidium and Basidiospores. Here also it will have ascus and ascospores. Okay, so here you might have seen this, okay, while studying in some other topic, bracket, which is a sexual fruiting body in Ganoderma lucidum also. Okay, tangible, you might have seen this type of structure at the base of the infected tree. There is a uh, mushroom shaped, okay, bracket shaped structure, ear like structure, which is nothing but Basidium, which is otherwise called bracket, which is otherwise called sexual fruiting body produced by the Mycota. Okay, so under the surface of the Mycota, it will be having minute openings, which will releases Mycota. Okay, so this bracket is related to the Mycota. The rest all the 3 is related to the Scomycota. So the correct answer will be the bracket because they have asked about the incorrect answer. Okay. Next, I'll read the question. In rice, a night temperature of 20 degrees Celsius alternating with day temperature of 30 degrees Celsius with 14 hours of light and 10 hours of darkness is most favorable for the infection by. I have already discussed related to this question. Okay. So the pathogen infecting in rice, a fungal pathogen infecting in rice called oryzae, which causes rice blast disease, which is more favorable at this temperature of 20 degrees Celsius. Okay. So it needs low temperature for its infection process. One more thing also we have discussed. For its infection process, it needs melanin as a constituent for penetration and infection process. That's why we are using the melanin synthesis inhibitor as a fungicide to reduce or to cut off the infection process. Okay, so here the correct answer will be the Magnaporteveraceae, which causes rice blast, which requires very low temperature for its infection process, which will be more favorable for the infection. Okay, next, the technique shoot tip culture is employed to get. The answer will be virus free plants. You might be knowing the shoot tip, which is nothing but, so when we take a plant, the topmost portion will be the shoot tip, okay, which is also called as meristem tip also we can say. Why we can use the meristem tip for virus free plants? There are so many reasons for selection of such meristem tip or shoot tip to get or to attain the virus free plants are. There are many reasons. Okay, more important reasons are there will be more oxygen production at the shoot tip area. The next reason will be uh, it is said that the plasmodesmata, which is cell to cell connection, will be not much developed at the meristem tip area or shoot tip area. So even the virus cannot able to penetrate into the cells of meristem tip area. Okay. And one more important reason is there will be more fast development at the meristem tip area. Okay. So based on all these reasons only, there will be no or very less infection of virus will be there at the meristem tip or shoot tip area. So to get a virus free plant, scientists always choose the meristem tip area so that they can culture it to get the callus, then mass multiply it using the tissue culture technique. Okay. The next question. The polymerase chain reaction technique, simply called PCR, was developed by, the answer will be the Kary Mullis. Okay, so yeah, you might be knowing that the technique related to the PCR, which has the three important steps: denaturation, annealing, extension. Okay, so denaturation is a step which is unwinding. Okay, so the double strand will get single stranded, then annealing is nothing but primer will get attached, then nucleotides will be added in a linear fashion. Okay, then extension means synthesis of new strand. So the again the process will get repeated. So only three important steps will be there in the PCR process: denaturation, annealing, and extension. So using this process, we can multiply the DNA strands, or a particular strand of the gene also we can able to multiply in a multiple times. Okay, so which was developed by the Caramellis, so that he was awarded Nobel Prize for the chemistry in the year 1993. He was developed this PCR technique in the year 1983. After 10 years only, he got the Nobel Prize in the year 1993. Okay, the next question. So definitely one question will be there related to the deficiency. Okay. Here, the whiptail of cauliflower is due to the deficiency of, obviously, molybdenum. Okay. So here you can compare the healthy, or more related to healthy. And when you compare to the left, extreme left, there will be whip-like structure. Okay. So prolonged reduction of the leaf lamina, which is called whiptail of cauliflower, more related to the deficiency of. Now, leaf blotch of turmeric is caused by. They have asked about the disease, and we have to choose its scientific name. Okay. So, leaf blotch of turmeric is caused by Taphrina maculans. One more question also you might have heard: Taphrina deformans, which causes peach leaf curl. Okay. So, one more disease also now you are learning. Leaf blotch of turmeric is caused by Taphrina maculans. The next question is: wilt case is common in Dobshness, blast of paddy, bacterial blight of paddy, white rust of crucifers, red rot of sugarcane. Okay, so blast of paddy you might know, related to the Magnaporthoraceae fungus, which causes spindle or eye-shaped lesions, which coalesce and causes a drying of the leaves, and white rust of crucifers, which shows symptom called stag head symptom, white blisters also we can say, which is caused by an oomycete called albugo candida or albugo blighty. Then red rot of sugarcane is due to a fungus called Colitotrichum falcatum. Okay, here the answer is bacterial blight of paddy. Okay, how I can say? So in bacterial blight of paddy, there are, we can say there are of two phase. Okay, in nursery or early stage phase, it is showing wilting phase, which is otherwise called crescent phase symptom. And on the old age phase, otherwise after transplanting, it will show blighting phase. Okay, leaf blight phase. So in bacterial blight of paddy, there are of two phase. Young age seedlings, it will shows crescent or wilting symptom. After a certain period, okay, it will shows leaf blight phase. Okay, they might even ask, crescent phase is also common in bacterial leaf blight of paddy. Okay, so you should know both the terminologies: wilt phase, otherwise called crescent phase, which is must knowing question. Okay, important question also. The next question: green ear of Bajra is caused by, so green ear, which is related to the downy mildew, Sclerospora graminicola. Okay, so downy mildew of Bajra is otherwise called green ear, which is caused by Sclerospora graminicola. The term green ear is nothing but the ear head, the grains will be converted into leaf-like structure. So we can say as a green ear, which is caused by a oomycete called Sclerospora graminicola, which is a downy mildew organism. Okay. So one of the most important question, an important disease infecting chili fruit is obviously anthracnose. So not only in the chili fruit, even in the fruits also, fruit crops also, wherever there is sunken lesions, in that sunken lesion, when you see the concentric rings, which is nothing but fruiting bodies called acervali. Okay, so they are all caused due to anthracnose only. So anthracnose also we can relate with the latent infection. Okay. So you can even study it in mango, guava, papaya. Okay. So most of the post-harvest diseases also will be related to the anthracnose only. Okay, and in banana also it is most severe, and in chili fruit it is most important disease in the case of chili, and it will not only affects the chili fruit. In the field itself it will show symptom, and plant it will show dieback and a leaf spot. Then in fruits also it will show such fruit infection, that is sunken spots. So the infected chili fruit will be loss of pungency and flavor will be get lost. And most important thing is it not only affects the surface, the fungus will also penetrate inside the fruit and affects the seeds also. So which causes externally seed borne pathogen. Okay, so we should not use the seeds which is infected by the anthracnose, because the seeds also may contain the or adhered with the pathogen will get adhered with the seeds. Right. Next, the major diseases of sorghum crop are, okay. Here itself you can guess. They have asked in a plural form. Okay, diseases are, so these are all the important diseases affecting sorghum. Okay, grain mold, rust, and downy mildew. So the answer will be all the above. Sigatoka is the problem in. Okay, so the answer will be banana. Okay, so generally, Cercospora leaf spot you might have heard. When it affects the banana, we can say as a Sigatoka leaf spot. In banana, you will have significant names for each diseases. Okay, for example, for fungal wilt, we can say as a Panama wilt. For bacterial wilt, we can say as a Moko wilt. Okay, for leaf spot, we are saying as a Sigatoka leaf spot. Okay, okay. For banana black mosaic virus, we are saying as a Kukan disease. Likewise, for each diseases in banana, we are giving a specific name for the disease. So, for such leaf spot disease, infecting in banana is Sigatoka leaf spot. Okay, there will be elongated or elliptical leaf spot of ash-shaped ash color with grayish color with brown margin. Later on, each spots will merge and will coalesce, dry, and will lose in yield. Then bacterial blight of rice is caused by, okay. So before two questions only, we have discussed about the bacterial blight of rice, which shows two different type of symptom. During the young stage, it causes crescent symptom, and in the after the a certain type of aged seedling, it will show bacterial blight symptom, which is due to a bacteria called Xanthomonas oryzae, pathovar oryzae. Okay, XOO, Xanthomonas oryzae, pathovar oryzae. Next, match the following using the codes. Okay, so you must know the disease and its causal organism. Okay, at least which type of pathogen it is. Okay, so only when you are knowing this type of uh, disease and its pathogen, then only you can match this. So, citrus canker is due to bacteria called Xanthomonas citri. Lead blight of potato is due to fungi or oomycete also we can say. Okay. Phytophthora infestans. Banana bunchy top is nothing but virus. Banana bunchy top virus. Okay. Red rust of tea is algal disease. Okay. Cephalurus. Okay. Not only in tea, in guava or some other fruit crops also it will infect a red rust, which is an algal disease. So rust is there. A red rust is different, general rust is different. So whenever they are asking about the red rust, you must click the option as algae only, which is not a fungus. Okay. So for confusing only, they have given fungi also in the option. So without knowing the difference, you should not choose. So whenever they are asking as the red rust, which is definitely a algal disease only, not the fungus. So the correct answer will be the option A. Next, the major approaches in plant disease control are, so generally, there are six principles in plant disease control: avoidance, exclusion, eradication, protection, resistance, and therapy. So they have given three from the six principles. So the answer will be the all the above. And the next question is the antagonistic fungus used in crop disease management. Okay, here you must observe about they are asking about the antagonistic fungus and crop disease management. Okay. Now the answer will be Trichoderma, because Trichoderma is a fungus which is used to reduce the pathogen incidence. Okay, it will fight with the pathogen fungus. Okay, a fungus which is targeting on pathogenic fungus. Okay, so that's why it is called as antagonistic fungus. This Trichoderma is an antagonistic against the plant pathogen. Okay, for confusion only, they have given trichogramma also. Trichogramma is also a bioagent which is used to control the insect pest. Okay. So you must know the difference between the trichoderma and trichogramma. So choose the option wisely. Then aphids are the vector of. Okay. So I have already discussed related to the vectors. Now they have given the option as banana bunchy top, brinjal little leaf, cardamom mosaic, cowpea mosaic. So here they have given option as choose one, two, three, like that they have given. So you must know all the three, four answers also. Then only you can pick which are all the diseases which is spreads through the aphids. Okay, so we can eliminate few like brinjal little leaf, which is a phytoplasma disease. So phytoplasma disease always or mostly they are spreads through leaf hoppers only. So we can eliminate this option. Okay. So B will not be there in our answer. Okay. Here we'll come to the A option. Banana bunchy top. We have already discussed about the disease banana bunchy top virus, which spreads through the banana aphid or black aphid, Pentalonia nigronervosa. So definitely A will be there. Then I have already discussed in one slide related to a one question. Wherever you are seeing mosaic related viral diseases, definitely most of the mosaic diseases will be related to the aphid as a vector. So cardamom mosaic virus also spreads through aphid. Cowpea mosaic virus also spreads through aphid only. So the answer will be A, C, and D. So the correct option will be C. That is 1, 3, and 4 will be the correct answers, which are the vectors for the as aphids. Okay. And the next question, rice tungro virus is transmitted by, the answer will be green leaf hopper. So definitely there will be a question will be coming related to the vectors. Okay. So rice tungro virus is transmitted by green leaf hopper. They may even ask related to the phytoplasma diseases also. Okay. Little leaf of brinjal. Okay. So which is due to the leaf hopper Hishimonas. If it is like that, not there also ask. Okay. Sesame pylori, which is also transmitted by the leaf hopper. Orochius albicindus. So all the viral diseases and its vector you must be knowing. Okay. And here, next question. Just now we have discussed about this. Sesame pylori is caused by phytoplasma-like organisms. First of all, I'll give a brief related to the plant pathogens. Okay. So first one is protozoa. You can even give an example of clubroot of crucifers. Okay. Next, fungi, bacteria, and here the bacteria will be we can say as an example of bacterial leaf plate also or citrus canker also. So it will be present on the surface of the plant system. Certain type of bacteria will be present only inside the xylem and phloem vessels of the plant, which is called fastest vascular bacteria. Certain type of bacteria will be wall-less in nature, or okay, so it will not have definite shape. That's why it is called molecules. So in the molecules or wall-less bacteria, there are of two classes or two classification: phytoplasma and spiroplasma. Spiroplasma will be in spiral shape. Phytoplasma will not have spiral in shape. Okay. Then later on, very minute than the phytoplasma and spiroplasma is virus. Okay, which is next one is vira, which is smaller than the virus. Then certain apart from the minute organisms, algae is also a disease. Algae is also an organism which causes disease. Then phanerogamic parasite also we have discussed. Then abiotic disorders. We should not say as a disease, which is a disorders. Okay. So Sesame pylori, which is a phytoplasmatic organism. So phytoplasma. Okay. Next. Which of the following is a vector of bendy vein clearing disease? So most of the question will be related to the viral diseases and its vectors only. So you must be very aware about the viral disease and its vectors. So here I have already discussed about this bendy vein clearing, which is otherwise called bendy yellow vein, okay, mosaic virus, or otherwise called bendy vein clearing. So which is an important disease which spreads through whitefly. Okay, then this one also we have discussed already. Loranthus is a partial stem parasite. So we have already discussed this flowchart: phanerogamic parasite, stem parasite, and root parasite. In both of the stem and root parasite, there are two subclasses: total parasite and partial parasite. So total stem parasite, partial stem parasite, total root parasite, partial root parasite. So each of the parasite along with its scientific name and its host, you must be much aware, then only you can give the correct answer. So Loranthus here is a partial stem parasite. Why? Because partial stem parasite, because it contains few leaves on its own. Loranthus will be having some leaves, so it will derive some of its nutrients on its own, and it will absorb some few more nutrients from its host. Okay, so that's why it is called partial stem parasite. Then the symbiotic association of fungal hyphae with plant roots is known as mycorrhiza. Here, when you split the term mycorrhiza, it is myco means fungus, rhiza means root. Okay, plant root. So here itself we can uh, opt the option. Okay, fungal hyphae myco, plant roots as a rhiza. So mycorrhiza will be the correct answer, which is the association between the fungus and the plant roots, which is called mycorrhiza. Okay, so this is about the discussion related to the previous year examination questions related to the plant pathology asked in agricultural examinations. Okay, hope you understand and you have cleared much more doubts, and you have gained some more knowledge apart from the questions they have asked in the examinations. Thank you so much for watching. Stay tuned.